Thank you. And this is joint work with Chris Wallace. I first want to briefly go over genome-wide association studies and the types of data we can leverage to improve power in these studies. And so in a GWAS, we measure a collection of variants, and we want to test for association between that variant and, in this case, a rare disease of interest. And specifically, we're going to focus on single nucleotide polymorphisms. So these are base pair mutations that occur in at least 1% of the population. And they're coded numerically 0, 1, or 2, and it's just the number of minor alleles that individual contains. And we know that along the genome, where the mutation occurs and the genomic context, I'll go over in a second, matters when trying to understand its effect on that rare disease. We also know that most variants are pleiotropic, and they actually impact multiple traits simultaneously, for example, through a shared biological pathway. And so the first source of data we can think about is trying to identify genetically related traits and trying to leverage that to improve power in the study we care about. The second source of information are genomic annotations. And this is trying to leverage all the data we've accumulated to this point, trying to characterize the variant. For example, is it a promoter enhancer region? Is it genomically constrained region? Can we leverage that information to not only improve power, but to better prioritize variants? I'm going to focus most of the talk on genetic pleiotropy, and towards the end, I'll go over some recent stuff on genomic annotations. Okay, so I'm just going to go over kind of how we analyze this and how we can do this. So in a GWAS, we measure SNPs, and we test for association between the trait, and we calculate our test statistic and corresponding p-value. And once we do that, we then select a genome-wide significance threshold to control the family-wise error rate. And we say everything below that is statistically significant. Now, we know what determines our power in the study is the sample size. But for rare diseases, it costs a lot of time and money. So this is often a constraint. And so what people realize is, well, let's try to leverage pleiotropy from other related traits. And so as a toy example, if body mass index was a trait we're interested in, we may try to leverage body fat percentage, cholesterol, and triglycerides in order to improve power in that study. And so what our problem looks like is when we go SNP by SNP, we not only want to consider that p-value, we want to consider the summary statistics from all those genetically related traits at the same time. And the way we're going to do this turns out through a false discovery rate framework. And before I get into it, it's worth going over different measures of significance for variant discovery. And so we're trying to test the null hypothesis, which the variant is not associated with the trait, versus the alternative, which it is. And we typically calculate a p-value to control the family-wise error rate. And we know the family-wise error rate is usually too conservative. So there's other measures out there. One's the local FDR, and this is just the Bayesian classification error rate. It's the probability of the null given the data. And it's viewed as a local assessment of significance. The other measure is the Q-value. Now, the Q-value controls the false discovery rate, the expected proportion of false discoveries, and it's the minimum false discovery rate or positive false discovery rate incurred when calling a set of tests significant. And this is a global measure of significance, and it's commonly used in genomics. So what we want to do is estimate these significance quantities while integrating that SNP level information. Okay, so let's go over the intuition of how this is going to work. So on the y-axis, we have the p-values from our study. And on the x-axis, we have the summary statistics from a genetically related trait. And so we know the smaller the p-value, the higher chance is coming from the alternative, and the higher the p-value, the higher chance it's from the null. And then similarly, these are p-values on the x-axis, so it's going to mirror it. So in a standard GWAS, we fix that significance threshold and say everything below that is statistically significant. But if you're leveraging a genetically related trait, this significance region is going to be nonlinear. Okay? So we want to identify a function that's going to be a function of our p-values and our SNP level informative data. We want to try to figure out what function it should be. And fortunately, it's been figured out before. This is John Story, 2003, figured out that the local FDR minimizes the Bayes error rate. And I'm going to call this the functional local FDR because it's the function of our informative data. And so our strategy should be based on this function. And we're going to extend a procedure to estimate this from John Story's group. Okay. So as I mentioned, this is our significance region. It's our decision rule. We're going to make, now we want to calculate the functional Q-value and P-value. And so the functional Q-value is the Q-value. Now it's a function of the informative level data. It's really easy to calculate. It's just the average of the local FDR in the region. Now, the thing, we're introducing the functional p-value. All it is a mapping back to a p-value, but now we've incorporated that informative level data. And I didn't introduce pi naught, but it's just the prior probability of the null, and it's a special quantity in the multiple testing space. Now, if you work in genomics, you usually just have Q values and you're done because you're controlling the false discovery rate. But in GWAS, we really care about the family-wise error rate. And so it turns out to be easier to incorporate it when calculating the Q values and then actually mapping it back to this p-value quantity. And everything on this slide just depends on estimating the functional local false discovery rate. And so that's our estimation goal. Okay, so our framework we're calling surrogate functional false discovery rates. It works as follows. We take our data and we're going to split it up into a set of LD independent SNPs and a set of dependent SNPs. Now with the independent SNPs, we're going to estimate the local FDR, which depends on two quantities. The numerator is the conditional probability of the null given the informative data we have. And so we can get that using a GLM, a GAM, and there's a lot of sophisticated things you can do. The denominator is a little trickier. It's the conditional density of the p-values given the informative data. And we want to have a non-parametric estimator. And non-parametric density estimation is quite hard in higher dimensions. And so we construct a surrogate variable based on pi naught to keep it as a two-dimensional problem. And we find this works out really well in practice. And so once we have these fitted curves, we can go back and then predict the local FDR of the left out SNPs. And as I mentioned, once we have the local FDR, we can get our Q values for FDR control, and we're going to map it back to this functional p-value quantity to control the type 1 error rate. And so that's just a brief overview of the method. Okay, so next we wanted to evaluate SIFTER using the UK Biobank. So we took the UK Biobank and we split it up into two different data sets. One where the primary trait we're interested in was BMI, and the other were the informative traits for body fat percentage, triglycerides, and cholesterol. And then we downsampled BMI to different sample sizes. And we wanted to ask what happens to the functional p-values compared to the regular p-values here. And here we have a plot on the y-axis of the number of discoveries. On the x-axis is a function of downsampling proportion. And in gray are the standard p-values, and in black are the functional p-values. And we see across all downsampling proportions, we find an increase using the functional p-values. And so something we've kind of already known, which is when you leverage related traits, you can substantially improve your power. Now, then we asked of the new discoveries made by SIFTER, how many overlapped with a meta-analysis with the left-out data set, and we find nearly all the discoveries overlap. And then we wanted to ask, well, how does it translate in terms of the number of independent loci, so not including the LD SNPs, and we find similar results that it's increasing the number of independent locations, so it's not just inflating an LD region, which is good. The next question we asked is, well, how many additional samples would you need to gather in order to match the improvements in power? And as a function of downsampling proportions, about 60%. And so perhaps the important setting is, well, what if you use non-informative traits? So we permuted the non-informative traits, the informative traits, so it's uncorrelated with the primary trait, but we wanted to maintain those between trait correlations. And so when you compare the functional p-values, it should be approximately the standard p-values. And we see as a function of downsampling proportion, there's a strong correlation. And in fact, it tends to be a little conservative due to primarily estimation. The next thing we wanted to do is add null chromosomes to the original data. So we permuted the primary trait, and we randomly added null chromosomes to the original data. And so at the null chromosomes, the functional p-value should be approximately uniform. And when we do that, on the left-hand side, we have the regular p-values. On the right-hand side, we have the functional p-values from SIFTER. And we find that there are well-behaved p-values at the null chromosomes as well. And so SIFTER is not systematically inflating the type 1 error. Okay, so a really good application, we think, for this method is a rare disease GWAS. And so we applied it to eosinophilic granulomatosis with polyangiitis, EGPA. It's an extremely rare disease. It's characterized by inflammation of the blood vessels. Not much is known about it, but patients tend to have a history of asthma and high eosinophil count. So we have a GWAS, about 700 cases and 7,000 controls. And we're going to use clinically relevant traits of eosinophil count, childhood onset asthma, and adulthood onset asthma. And so we're going to apply SIFTER leveraging these genetically related traits. So first, we wanted to answer the question, what if we just throw in the non-informative data in this rare disease study? You should approximately get the p-values of where there's signal. And we see as of on the y-axis, we have the functional p-values. On the x, we have the regular. And it's approximately the same, and particularly where there's actually signal. So that's good. So to go over the results, in the original GWAS, there's 2 loci that are genome-wide significant, and there's a few others that are pretty close. When we apply SIFTER, we identified 8 additional loci, and we find that they're all involved with they're near genes involved with immune-related processes, and they overlap with the 4 traits, which is good. That means, they're the regions that are being informed by the data. So now I'm going to switch gears a bit and talk about more recent work on leveraging genomic annotations. So as I mentioned, where the variant is in the genome plays an important role in understanding the disease, right? So what's its role in gene regulation? Is it a promoter-enhancer region? How close is it to the transcription start site? Is it in a genomically constrained region, meaning it doesn't vary across species, which implies some functional importance, perhaps? And other LD-related quantities, such as recombination rate and other population genetic quantities like nucleotide diversity. And we're going to use a common model used in this field. It's called the baseline LD model. And it's a collection of 97 annotations that are high quality. And we're going to try to use these scores within our method. And so we're going to evaluate the LD scores using the UK Biobank. First, I just want to give you a brief kind of intuition of what these baseline LD scores are doing. If we had an LD region, and this is a region of correlated SNPs, and you had a single causal SNP, and nearby you had a non-causal SNP, and they're both significant, the LD score really tries to partition the signal across the various annotations. So AJK is the annotation, and scale it by the correlation. And so this equation doesn't come out of thin air, it comes out of some algebra. But we found that when we use those in SIFTER, it does a lot better than using raw annotation values. So we then took the UK Biobank and we focused on 27 independent traits. And we wanted to compare the p-values to the functional p-values from SIFTER, leveraging these baseline LD scores. And what we find is that in terms of the total number of discoveries across the various classes, that SIFTER, the functional p-value increases the power. And then we asked, well, in terms of the number of independent loci, which we define conservatively in this case, we also observe an increase as well. And so kind of what we know, previous work has also shown that these baseline LD scores can really improve common variant discovery. But what we were really interested in was trying to evaluate annotations or predictions from a new class of models called foundational models. And this is work with Josh, John, Sash, and Chris. And so what a foundational model is, it promises to provide a direct effect size basically at each variant site. And so I'm not going to go over the deep learning algorithm, but you input a variant sequence, and it's going to output predictions of different data modalities. For example, RNA-seq, DNAs, histone modifications. And so remember, annotations are a proxy for function. And so these models really promise to provide basically an effect size of what happens when you have that mutation. So what we wanted to ask was, do foundational models provide any unique information than what we already know? And so we're going to add to the 97 baseline LD annotations, we're going to add a foundational model called Borzoi from Calico Life Sciences. And the figure from the left is from Alpha Genome. We're working right now to get those annotations, so these are preliminary results right now. And they released a set of 100 variant scores that they found perform well in functional fine mapping and other stuff. And so when we applied it to our data set, we found mixed results, maybe a slight increase for disease-related traits. And in terms of the independent loci, there's also mixed results. And so one thing to keep in mind, this is common variant discovery. And so usually these models tend to be evaluated in the rare variant space. And so while Borzoi does not yet lead to substantial improvements, it's not to say future iterations, as it's trained on more and more data, will provide better predictions. Okay, so just to conclude, we have a framework to incorporate SNP level data. Depends on estimating this local FDR statistic. But once we have that, we can get our functional Q values and this functional p-value quantity to control the family-wise error rate. We found it increases power and controls the FDR and type 1 error rate. And we get approximately the raw p-values back when we condition on non-informative data. We found that we enhanced variant discovery when we leveraged pleiotropy in a rare disease GWAS. When we leverage baseline LD annotations, we also find an increase in power as well. When we try to evaluate this new class of foundational models, we have mixed results at the moment. And so with that, I want to thank Chris Wallace and the Wallace Lab and our collaborators, Josh, Sash, and John. And the first part of the presentation is recently published, and we have an R package you could use. Thank you.